Hello, my name is Eric Letouzé. I am a computational biologist at the Cordeliers Research Center in Paris. And today I'm going to show you how, by combining whole genome sequencing and Bionano genome imaging, we could unravel complex structural rearrangements induced by replication stress in liver cancer. In the lab, we are interested in liver cancers, and in particular, hepatocellular carcinomas, which is globally the third cause of cancer death. This cancer has a heterogeneous incidence in the world related to the diversity of risk factors. Most cases occur in sub-Saharan Africa and Southeast Asia related to HBV infection and aflatoxin B1 exposure. There is also a high incidence in Japan and Egypt due to HCV infection. In France, alcohol is the main cause of the disease. And in the US, metabolic diseases are the principal risk factor. These risk factors operate by damaging the liver. And it is known that more than 80% of HCC develop in a cirrhotic liver by the transformation of dysplastic nodules. However, a little less than 20% of the cases develop on a non-cirrhotic liver. In this case, one of the main transformation mechanisms is the activation of oncogenes by viral insertions or structural rearrangements. In particular, we recently described a new molecular entity of HCC driven by viral insertions and rearrangements activating cyclins A2 or E1. We found that tumors with activated cyclin A2 or E1 define a homogeneous molecular group of HCC, as you can see on these gene expression-based classifications. They also have specific clinical features, in particular a poor overall survival. There are also large tumors, usually bigger than 10 centimeters, occurring in relatively young patients and in absence of fibrosis. At the molecular level, these tumors, called CCN-HCC, display a high frequency of RB1 and PTEN inactivation, as well as a striking activation of ATR pathway implicated in replication stress response. Indeed, cyclins E and A are implicated in the entry and progression into S phase. And it is well known that when you activate cyclin A2 or E1, you will induce premature S phase entry and replication stress. Then we wondered what kind of genomic instability could be induced by this replication stress phenotype in this tumor subgroup. So we sequenced the whole genome of 50 tumors comprising CCN-HCC and tumors of other molecular groups. We found no difference in terms of point mutations and indels, but we found that CCN-HCC display a very high number of structural rearrangements. In addition, when looking at the nature of these rearrangements, we found that replication stress in these tumors induces a specific signature, the RS1 signature, which is characterized by focal tandem duplications coming together with a lot of translocation breakpoints. This phenotype is easily recognizable on a circus plot. Here you can see the abundance of tandem duplications in green and interchromosomal translocation breakpoints in gray. And it also leads to a particular copy number profile with hundreds of focal gains distributed throughout the genome. To get a deeper understanding of this phenotype, now we can zoom on one particular chromosome. Here we are. And now we can see in more details those very focal gains that are very clear on the log ratio profile. And on top of this log ratio profile, we overlaid the abnormal junctions identified by short-read whole genome sequencing. In some cases, the focal gains are induced by classical tandem duplications, leading to the duplication of one segment adjacent to itself. But in other cases, like here, a focal gain is actually surrounded by interchromosomal translocation breakpoints, here going to chromosome 2. And if you go to chromosome 2, you will see another very focal gain with interchromosomal breakpoints going back to chromosome 14. This kind of event, typical of this tumor subgroup, is called templated insertion cycles and is characterized by many focal gains being interlinked with interchromosomal translocation breakpoints. Now, it's very difficult with short-read whole genome sequencing to figure out precisely what this complex event looks like. All we can do is make hypotheses, and we found two hypotheses that could explain this kind of observation. One in which the E segment would be inserted between two copies of the B segment inside chromosome 14, and another equally likely possibility in which the B segment would be inserted in the middle of two E segments on chromosome 2. And because there's no way with whole genome sequencing to demonstrate the existence of these events and to discriminate these two possibilities, we decided that we would analyze a few cases with Bionano. So we analyzed 4 HCC samples and matched normal liver with Bionano optical mapping technology, starting from 30 milligrams of frozen tissue. We successfully obtained high molecular weight DNA, more than 150 kb long, for all but one sample for which we did not provide enough starting material. We obtained a genomic coverage around 300x with an average molecule size of 250 kb. So this was a very important step, and we were happy to obtain, to be able to extract really long DNA molecules, because with our classical DNA extraction kits, we usually get DNA fragments of 10 kilobases. From this data, we analyzed structural variants using Bionano software. And because we are interested in the somatic structural variants that are specific of tumor cells, we made a detection in both the tumor and matched normal sample. And we compared the two SV call sets to be able to extract only the somatic structural variants that occurred in tumor cells. We then represented the global profiles of structural rearrangements with circus plots, as you can see here, for the 4 tumors. The 2 tumors on the left are CCN-HCC tumors driven by HBV insertion in cyclin E1 for the first and cyclin A2 for the second. And the 2 tumors on the right are tumors without activation of these cyclins. What we can see here is that we are able, with Bionano technology, to retrieve our RS1 signature of replication stress with plenty of tandem duplications and interchromosomal translocations. Interestingly, in the second sample, we knew that we had some tandem duplications that were very focal in the range of 10-20 kilobases. And for these events, usually you don't have enough DLE markers to be able to map with precision the segment on the genome. Yet, Bionano software did identify those events as insertions, despite their very small size. So the RS1 signature is detected exclusively in the 2 CCN-HCC samples. Next, we compared the structural variants called with whole genome sequencing and Bionano for 3 tumors that were analyzed with both technologies. Whole genome sequencing was performed as quite a high depth because we had a 90x coverage in the tumor and 30x in the normal samples. On this slide, you can see the number of structural variants per category identified by whole genome sequencing on top and Bionano at the bottom. The first tumor on the left is relatively quiet in terms of structural rearrangements, whereas the 2 others correspond to cyclin HCC. What you can see overall is that we have a very good match between the two technologies, with the majority of structural variants identified by both. However, for each tumor, we have some events identified only with Bionano and whole genome sequencing. Globally, we have more events identified with Bionano, on average 1.5 times more events than with whole genome sequencing. And we also have a larger diversity of events, including some intrachromosomal rearrangements. And we also noticed that some of the tandem duplications are classified as insertions. These correspond to the very small events. So we had a look at the events that were found only with whole genome sequencing, and they generally consist in very small events, smaller than 10 kilobases. And then we had a look at the events that were only detected with Bionano, which you can see on the next slide. Here, we have the coverage log ratio profile of one chromosome that is represented. 1 point corresponds to 1 kilobase. And on top of that, we overlaid the structural variants identified by whole genome sequencing on the top and Bionano at the bottom. We can see that many events are only detected with Bionano. These include centromeric events. It is known that these regions contain repetitive elements and are difficult to analyze with sequencing technologies. Others correspond to subclonal events that we detected due to the very high coverage we had in Bionano data. And finally, a lot of events correspond to highly complex rearrangements that were missed with whole genome sequencing approaches. Here, the red arrows, for example, point at a series of intrachromosomal rearrangements that bring together distal junctions and lead to a very complex rearrangement. And most of these junctions were missed in short-read sequencing. So now let's go back to our typical events induced by replication stress, which we wanted to characterize with optical mapping. Like I said, for this kind of interchromosomal templated insertion cycle, we cannot really detect the precise event with whole genome sequencing. All we can do is make hypotheses. In this case, there are two hypotheses, either segment B within 2 copies of segment A or segment A within 2 copies of segment B. With Bionano, we could obtain long molecules spanning the full events. So on this graph, you can see the molecules at the bottom and the map reconstructed from these molecules in gray. And if we go through this map, we can see that it starts with the first segment of chromosome 3. Then we have the B segment that is duplicated at 2 positions. And in the middle of these 2, you will see the A segment from chromosome 1, and then the end of chromosome 3. So here we can confirm for the first time the existence of these templated insertion cycle events, and we can identify unambiguously the host chromosome, in this case, chromosome 3. We also identify other kinds of events which are strongly related, but involving only intrachromosomal templated insertion cycles. In this case, we have 2 gained segments linked by inversion breakpoints. And with Bionano, we identified long molecules validating the event in which segment A is inserted into 2 copies of segment B at the end of chromosome 10. Finally, we could reconstruct very complex events involving several segments, like this one involving 3 segments on chromosome 3 and 1 segment on chromosome 6. With Bionano, we could unambiguously identify the complex event that involves the insertion of segments C, A, and D in the middle of 2 copies of segment B located on chromosome 3. So overall, we have a diversity of structural rearrangements related to replication stress. About half of them are classical tandem duplications, and another 40% are templated insertion cycles that can involve intrachromosomal or interchromosomal events. All these events that represent 82% of alterations in this subgroup can be explained by a break-induced replication mechanism in which the replication machinery is stalled at some point, will jump to another location, replicate a stretch of DNA, and finally come back to the original site of replication. In conclusion, we identified a subgroup representing 7% of HCC, driven by cyclin A2 or E1 activation, that will induce premature S-phase entry, replication stress, fork collapse, and this particular signature induced by break-induced replication repair. This gives rise to large tumors, very aggressive, but this replication stress phenotype suggests these tumors could be targeted by PARP inhibitors or ATR pathway inhibitors. In terms of what Bionano helped us to do here, it allowed us to reveal more structural rearrangements than whole genome sequencing, to unravel complex events induced by replication stress, and it may be used to identify replication stress in clinical samples. I will finish by thanking my sponsors, my colleagues in Jessica Zygmunt-Ross's lab, in particular Quentin Bayard, who was the main PhD student involved in this project, and our collaborators abroad. Thank you for your attention.